Hello friends, this is Shubham and today I'm going to talk about the very basic concept in physiology, cell membrane. Actually the cell membrane is also called as plasma membrane. It is the outermost layer of the cell which consists of the cytoplasm, mitochondria and other organelles as you know. But today I'm going to focus on the cell membrane. Cell membrane is the double layered structure. Actually it separates the extracellular fluid from the intracellular fluid. Okay, so it consists of the two major substances, that is phospholipids and the associated proteins. However, carbohydrates and some other lipids are also the constituents of the cell membrane and helps in making the cell membrane. But the two major portions are the associated proteins and the phospholipids. About 50% portion of the cell membrane is made up of the phospholipids and about 47% of the cell membrane are made up of the associated proteins and 3% are made up by the carbohydrates and other lipids. So let's talk about in detail that what are the functions of the cell membrane and what are the structures and what does they mean and what does this diagram mean, which I've drawn over here. Actually I've drawn the cell over here. This is our cell and this is the extracellular fluid which is aqueous, and this is the intracellular fluid, means inside the cell, the environment inside the cell. Here will be our cytoplasm, there is the cytoplasm, here will be our nucleus and other organelles, Golgi apparatus, rough endoplasmic reticulum, ribosomes, smooth endoplasmic, like that structures. But cell membrane divides or separates the two environments, extracellular fluid with the intracellular fluid. They both are aqueous. Okay, so as I have talked that they are made up of phospholipids and the associated proteins. So first let me talk about the phospholipids. Actually these structures which I have drawn over here, these are the phospholipid structure. These are the phospholipid structures. Actually these circular portions, these are the phosphates, and these lines, they are the fatty acids. So they constitutes of the phospholipids. If I will zoom this structure, for example here, I'm zooming this portion, this one phospholipid, then let's understand it more clearly that what are the phospholipids and what are the parts. This is our phosphate portion and this is the fatty acids. Actually this portion is the phosphate. This portion is the phosphate group, and these are the fatty acids. These are the fatty acids. Okay, now the interesting point is that these phosphate groups of the phospholipids, these are actually the hydrophilic part, means they are water loving. That's why, as I have mentioned, that these extracellular fluid and intracellular fluid, they are aqueous. So if you will look over here, if you will focus over here, the phosphate groups of the phospholipids are presenting and are situated towards the aqueous side, because they are hydrophilic, water loving. Because they love the water, they are hydrophilic, these phosphate groups. That's why they are pointing towards the aqueous solution, extracellularly and intracellularly too. Okay, so these are the phosphate groups, these are the hydrophilic or water loving, so these are pointing towards the aqueous side, extracellularly and intracellularly as well. Okay, now let's talk about the fatty acids, which are the hydrophobic, or you can say lipophilic. Okay, hydrophilic, these are the phosphate, which I have told that these are the hydrophilic, or at the same time you can say lipophobic. Okay, either these are the hydrophilic or these are the lipophobic, which means they scares of the lipids. They don't love the lipids. They don't want the lipids. So that's why they are far from the lipid side. These these sides are the lipid side. I will talk about that. But these are aqueous toward, because these are towards the aqueous side, because they are hydrophilic, okay, or lipophobic. Now let's talk about the fatty acids. Fatty acids are the hydrophobic, or you can say lipophilic. Hydrophobic. They're hydrophobic or lipophilic. Lipophilic. Okay. Means they scares of water or they love the lipids. Okay. So that's why if you will focus over here, that fatty acids are away from the water side or away from the aqueous solutions. Okay. They are just presenting in between the phosphate groups. Okay. So here you can understand that why they are called as hydrophilic phosphate groups, because they are towards the aqueous solutions, extracellularly as well as intracellularly, and the fatty acids, they are hydrophobic or lipophilic. They are lipid loving, not water loving, and the phosphate groups are water loving, not lipid loving. Okay, so these are the concepts about the phospholipids. This is structure. This is structure is phospholipids. Okay, I have just zoomed one part of the phospholipid over here. There are millions of phospholipids in the cell. Okay, over here. Here I have not drawn. I have just drawn the upper and lower side of the cell. Now, this was about the phospholipids. Also, one more important thing about the phospholipids, that these upper leaflets of the phospholipids of the phosphate groups and these lower leaflets of the phospholipids or these phosphate groups, they are just not the same or not identical to each other. They are the different, because the proteins attached to it are somewhat different, and also due to the property of fluidity of the cell membrane, they are not just identical to each other, or they are not the same, because these cell membranes and these phospholipids have the property to move along the cell. Okay, why they want the fluidity, or why they are mobile? Because they can move, because these cell membranes have to pass many substances through it. For example, very small molecules or very low molecular weight substances can pass through it easily. Very small molecules or very low molecular weight substances, or the substances which are lipophilic, they can pass through the membrane very easily. Whereas the water soluble substances or bigger substances, they need some passage to cross the cell membrane into the cell. Okay, so due to that, they are just not immobile. So the upper leaflets and the inner leaflets of the phospholipids, these phosphate groups, they are not just identical to each other, they are slightly different, because the proteins attached over the upper leaflets of the phosphate groups and the lower leaflets of the phosphate groups, which is inner side of the cell, are just different. Okay, so this was about the phospholipid structure. Also, you can see the stars over here which I have made. These are the carbohydrates. Because the carbohydrates are also attached to the upper leaflet of the phosphate groups, you can see these carbohydrates are attached to the upper leaflets of the phosphate groups of the phospholipids, and the inner one, they don't have the carbohydrates. So they are just present extracellularly, you can say, carbohydrates. Now, if we will talk about the carbohydrates, carbohydrates also have many important functions. One of the function is that they are present extracellularly with the phosphate groups of the phospholipids, which is also the cause that why these upper leaflets are slightly different from the inner leaflets of the phosphate groups of phospholipids. Second reason is carbohydrates, they are helpful in the cell adhesions. means they help one cell and another cell to adhere with each other, or the binding with each other. Actually, the they can bind to the glycocalyx of another cell. So glycocalyx of two cells, when they mate, they will cause the adhesion of the cells. So carbohydrates helps in this condition. Okay, this was about the carbohydrates. So we were talking about the fluidity of the and mobility of the cell membrane. They are just not immobile, they are moving along with the cell. They are moving along the cell in the cell membrane. The fluidity can be affected through some parameters, or by some parameters they can be affected. Now what are these parameters? For example, if the temperature, anyhow, if our body temperature increases, eventually these are the cells of our human body. If our body temperature increases, so temperature of the cell also increases, which will cause the fluidity of this membrane, which means the temperature, if the temperature of this cell increases, the fluidity and the mobility of the cell also increases. So temperature is directly proportional to the fluidity of this cell membrane, or phospholipids, you can say. Okay. Also, I have made these star-shaped structures over here in between the fatty acids, in between the hydrophobic or lipophilic parts, as I have already mentioned over there. Okay. So what are these structures? These are actually the cholesterol embedded within the fatty acid structures. These structures, they are embedded within the fatty acid structures, cholesterol. So actually, if there is more amount of, or if there is increased amount of cholesterol within the cell membrane, that will cause the lower or decreased fluidity of the cell membrane. Also, the membrane biogenesis, or the membrane fluctuations, cell membrane fluctuations, or cell membrane biogenesis, they can also affect the fluidity of the cell membrane. So the temperature, if it is increasing or if it is decreasing, it can affect the fluidity in the directly proportional manner. For example, if the temperature is increasing, the fluidity will also increase. If the cholesterol amount is increased within the fatty acids or the lipophilic or hydrophobic part, so the fluidity will be decreased. Okay. And if you will see that what structures are made up of these phosphates or phospholipids, actually extracellularly they are made up of the sphingomyelin or sphingolipids. Okay. And we will talk about the inner side. They are also made up of the substances. Okay, for example, phosphatidylserine, phosphatidylethanolamine, phosphatidylethanolamine and phosphatidylserine. These are the main substances which are present in the inner leaflets of these phosphates or phospholipids. Okay, and the sphingolipids or sphingomyelin, these are the main structures present over here, or phosphatidylcholine. These are the main phosphatidylcholine or sphingolipid or sphingomyelin. They are main substances present in the outer leaflets of these phosphate groups. So this was about the phospholipids structure. Now let's talk about also the fluidity depends on the some various factors. I have talked about the temperature, I talked about the cholesterol level, for example, or any other substance increase or decrease can affect the fluidity too at the same time. But you must have heard about the exocytosis and endocytosis in the cells. So this is also the phenomenon concerned with the fluidity of the cell membrane, because the cell membranes do move or do show the properties of the mobility. That's why the cell is able to show the properties of exocytosis, that is engulfing substance, or endocytosis. Okay, endocytosis is engulfing the substance from the outside, or exocytosis from the inside to the outside. So these are some parameters which affect the fluidity of the cell membrane. Okay. Now along with the phospholipids, along with the cholesterol, carbohydrates. Okay, just let me remind you what are these structures. These are the carbohydrates, carbs. Okay, these I have talked about that these are the phosphate groups, these are the fatty acids, this is the hydrophilic part or lipophobic, these are the lipophilic or hydrophobic parts. Same condition is over here. Now these star shaped structures, these are the cholesterol. Okay, these star shaped structures, these are the cholesterol. I'm making it over here. These star shaped structure, these are the cholesterol. Okay, which are also the important part of cell membrane. Okay, so what I have drawn over here, this structure, actually these are the channels. I have already told you that through these lipid biliary structure, because these are the double membrane structure, cell membrane or plasma membrane which surrounds the cell, what substances can pass through it? Okay, lipophilic substances, lipid loving substances. Okay, lipophilic substances can easily penetrate the cell membrane as compared to the water soluble substances. Okay, also very small molecular substances, or the substances, those substances which have very low molecular weight, they can easily penetrate or they can easily pass through the cell membrane or plasma membrane. Whereas some bigger structures, some water soluble structures, what about those structures? They also want to pass the cell membrane, they also want to penetrate the membrane, so they need some passage. Okay, and these structures helps those substances to penetrate the or to pass through the cell membrane. This I have drawn the one channel which have the tunnel in between it, the longer tunnel in between it. This is the tunnel, for example, and through this tunnel they can pass some charged molecules, some polar substances. Okay, some charged molecules they can pass. For example, here, this is the substance which is charged. Okay, this is the charged substance, and it wants to pass it, so it will go through this tunnel and will pass through this cell membrane or plasma membrane. So this channel have the tunnel, and some water soluble substances or some charged substances, some polar molecules, they can pass through this tunnel. So this channel is the passage for these kinds of substances. Okay, now this was about the channel. Now we have talked about the phospholipid structures. That cell membrane, let me repeat, the cell membrane, it consists of mainly two parts, that is phospholipids and the associated proteins. Also they are made up of in smaller portions carbohydrates, cholesterol, some other lipids, but the two major portions that covers the cell membrane constituents, that is associated proteins and the phospholipids. Phospholipids, I have already talked about that. Phospholipids, about the parts of the phospholipids and some fluidity property, and some cholesterol, carbohydrates, addition to these phosphate groups. So now let's move on to the associated proteins. How they works and benefit the cell membrane, or how they helps to cross the cell membrane some substances. For example, they have many functions. Associated proteins, actually this channel is also the part of these associated proteins. Associated proteins, they can be divided into two parts. Actually the integral proteins or the transmembrane proteins, which I have drawn over here in the shape of eye, just to remember it, that integral proteins or transmembrane proteins, that are present through the cell membrane. That's why they are also called as transmembrane protein or integral protein. Okay, they also have the carrier proteins. Okay, they also have the enzymes which helps in biotransformation of some molecules. Also they have the linker proteins. You can see over here the linker proteins. Okay, and they do have receptors. They do have receptors too. Over here I have drawn this structure for the receptor. So let's talk about these structures. First of all, let's talk about the integral protein, okay, or the transmembrane protein. What they will do, that some substances, this is the extracellular, okay, this is the extracellular, this is the intracellular, again this is the extracellular, just to not confuse you, okay. Now we're talking about the integral proteins, that some structures, okay, and by the way, these star-shaped proteins, these star-shaped structure which are drawn over here, as I told you, that associated proteins can be divided into two parts: integral proteins or transmembrane proteins, and the peripheral proteins, okay. So these are the peripheral proteins. They can bind in the inner side of the cell, and they can bind to the outer side of the cell with the phosphate groups of these phospholipids. Also they can bind to the integral proteins, okay. They can also bind with the receptors, for example 7-pass receptors or serpentine receptors, G protein molecule receptor. They can bind over there and can generate the first messenger system into the second messenger. For example, if any drug binds to the receptor, for an example, so if any drug binds to the receptor, it will generate the first messenger system, first messenger signal, and then that receptor, for example here, let's talk about the receptor first, that here is some drug comes over here, it binds to here, okay, so it generates the first messenger signal, okay, and it will show the signal over here, okay, it will show. So this is the, as it generates the signal by binding of the drug or any hormone or any other neurotransmitter to this receptor, they will generate any signal into the cell, intracellularly they will generate any signal. This is the second messenger signal. As they will bind, so that will the first messenger signal, and if they will show some response intracellularly, those receptor will show some response by the proper binding of the receptor to the any kind of drug or receptor or neurotransmitter or any other hormone, then they will show the second messenger. This one also helps in these associated, these peripheral proteins. These peripheral proteins are also attached over here. They can be attached over here. Okay, these are the peripheral proteins. So let me write over here that these are the peripheral proteins. So functions of the peripheral proteins: they can bind to these receptors intracellularly, okay, and can help the cell to generate the response, second messenger system, okay. Second, they can bind with the cytoskeleton inside the cell. For example, these are the structures. For example, I have drawn the cytoskeleton, okay. So these peripheral proteins helps in binding the cytoskeleton with the cell membranes, okay. So this is also the function of the peripheral proteins. Now let's move on to the integral proteins. Again, I have told you that the associated proteins can be divided into two parts: peripheral proteins and the integral proteins. Peripheral proteins I have talked about, that they could help in generating the second messenger signals inside the cell with the help of the receptors, any serpentine 7-pass receptor or G protein receptors, okay. They can bind to the cytoskeleton. Now if you will talk about the integral proteins or transmembrane proteins, okay. So if any signal comes, if any kind of hormone or any kind of protein binds over here. For example, insulin binds to it. Okay, insulin cannot cross the membrane easily, or insulin cannot cross the membrane on its own way, but they need some passage. Okay, so for example, insulin comes over here, they bind to here, they will generate the signal over here. Okay, they will generate a signal over here and will pass it to the inside the cell intracellularly. So this is the function of the integral protein. Okay, so now carrier proteins also helps to carry some proteins. They actually some, for example, over here it's a binding site, and some substance or some drug binds to it. Okay, and it will flip the site. So if it is like that and something binds over here, it will flip and pass the substance into the cell, and again it will flip back. So any substance will come over here, it will flip and helps the substance to pass into the cell. So this carrier protein works like that. Now enzymes, these are the enzymes which helps in biotransformation of some molecules. Okay, so enzymes are also present as far as associated proteins are concerned. Linker enzymes, they, remember I have told you about the cytoskeleton. These are the structure, these are the cytoskeleton. Okay, so these also, for example, here I have drawn again the cytoskeleton. These cytoskeleton, these are the intracellularly. And also you must have heard about the collagen fibers. Collagen fibers. So here are the collagen fibers, for example. Okay, these are the collagen fibers in the extracellularly over here. Okay, like these are the collagen fibers outside the cell. So these are the collagen fibers, and these are the cytoskeleton. So these linker enzymes, what they do, these linker proteins actually, these linker proteins will bind intracellularly to the cytoskeleton and will bind extracellularly to the collagen fibers. So these I have drawn over here. These are the collagen fibers. These are the collagen fibers. Okay, so these are the collagen fibers. So linker enzymes, what they do, they bind to the cytoskeleton intracellularly and they bind to the collagen fibers extracellularly. About the receptor, this is the receptor I have drawn. Let's put it R over here. Receptor, again I have told about this already, that our peripheral proteins can work over here, and our associated proteins also. This is the part of the associated protein receptor that any drug or any kind of neurotransmitter, hormones, or any other substance can bind to it, and if it shows proper affection to the receptor, okay, then it can generate the response, and the peripheral proteins can help in generating the response. So this was about the cell membrane, that about the phospholipid structures and the associated proteins, how they help the substances in passing through the cell membrane into the cell, and how they functions accordingly. So I hope this helps you a lot, and this was all about the cell membrane. I have discussed about the phospholipids and the associated proteins mainly, and in associated proteins about the peripheral proteins, as well as I have talked about the integral proteins or transmembrane proteins. So this was all about the cell membrane. I hope you like the video. See you in my next video. Thank you.